So today I'm going to be talking about playing with power. And this playing with power, I'm going to unpack for you in the next 45 minutes or so. And my goal today is to do a few things. First and foremost, make everybody curious. And next, to present this in the way where you may know a lot about physics and material science and robotics and go on a deep dive there, or you may know nothing and yet still really enjoy the topic and learn a lot. So we'll see if I can succeed with that. And let's take it away with actually a question for all of you. This may seem like a silly question, but it'll give me a sense of where you all are coming from and start to set the stage for some of the really, shall we say, non-intuitive aspects of the realm that I study. So we're looking at a cheetah running here, and I've given you some options of what is the fastest movement. And I love seeing all the answers coming in. Some of you might be not even happy with the question, what is fast anyway? Are we talking about speed, acceleration, duration? What is it? So, you know, the funny thing is nobody's chosen cheetah running. So do you know it's a setup? Someone should choose cheetah running. When I started this work over 20 years ago, the cheetah, and still today, of course, was the icon of fast movement, okay? And what I'm going to show to you today is this new world that we've discovered that really throws on its head even the notion of fast. So to give you a sense of the results, I'll put them up here. I think I have the power to do that. 37% chameleon tongue, jellyfish stinger, 39%, and squid strike, 24%. This is a very educated audience. Indeed, jellyfish stingers are the fastest yet recorded movements on the planet by an organism. And, you know, we're going to dig into this today, and I hope some of you are wondering what in the world I'm talking about, a jellyfish stinger. A jellyfish stinger is a single cell that is pushing out a tiny little needle that delivers the venom that gives you a jellyfish sting. And, you know, if you think about it, a jellyfish stinger at this tiny scale, moving one of the fastest movements ever recorded, is a totally different deal than a cheetah running. Why is it a different deal? Well, that's what we're going to talk about today. And we're going to address fundamentally, why is the cheetah so slow? Or why are other creatures so fast? And by the way, cheetahs are really fast. So I'm not saying they're not fast, but the world that we work on is, in my lab and with my colleagues, is a world of ultra fast, a world that we cannot see. We can see the cheetah, but basically it isn't possible to see with our naked eye any of these other movements that I'll talk about today. So the answers to that question invoke neuroscience, evolution, physics, material science, biomechanics. I've been working on this for over 20 years. I'm not even going to try to get into everything that we've discovered in this realm. Instead, what I'm going to do instead is try to bring to you this excitement, this feeling of how hard it is to get an intuition on something like a trapjaw ant strike or a mantis shrimp strike, and give you a taste of the promise that these systems have for the diverse capabilities in biomechanical systems, material science, and engineering that are not only become evident to us in the last 20 years, but is just completely accelerating at the moment in general as the field. But really, the answer to this question is summed up here, and I'm going to unpack it today. The fastest creatures have evolved exquisite control of mechanical power. Mechanical power is energy divided by time. So what these organisms do is they have incredible control of the duration and timing of energy release, such that, as I'll tell you today, a thing like a trapjaw ant, an animal like that, can close its jaws from the position that you're seeing right there with acceleration an order of magnitude greater than a bullet in a gun. And they can do it over and over again. They don't need to use gunpowder or anything. So what we're going to look at is how materials and structures in biology can do things that engineering can't currently, and to get a sense of how that's possible. So to get there, I just want to come back. I'm going to ask you just a couple more questions on the poll here, because a lot of what we talk about relates to scales and sizes that are not visible to us. So the first critter I'm going to introduce you to is an amphipod. It's a kind of crustacean. And the question here is, well, how does their size compare to a cheetah? And in particular, I want you to look at the, oh, come on, this cursor, there you go. Sometimes the, oh no, don't look. The cursor sometimes doesn't work on this. So the question here, right, is what does it look like? Okay, so what is the size of the little finger that the amphipod uses to close and snap compared to a cheetah? So the answers are coming in, and you all are like a good audience here. The vast majority coming in are right. The tiny little structure that this little crustacean uses is, in fact, the diameter of a cheetah's hair. So you can see the answers coming in here. But yeah, they're tiny. So let me show you on this, here we go. Let me show you on this slide what we're talking about. This little amphipod is, on the order of millimeters long, very tiny. The males have a claw that gets up to 25 to 30% of their entire body mass. And there's a little finger called the dactyl at the end of this claw that has the diameter of a hair. So imagine, if you have hair, you can take a hair and imagine moving it at the acceleration of a bullet in a gun. It's not possible to imagine that, which is my point in asking this question. The realm that we're talking about is unimaginable. And then how do we even understand why these organisms are making these movements, and how do they evolve the mechanisms to do so? So let's look at this funny little animal and look at the motion that it's making with that little dactyl. So this is a real-time video from some colleagues in Japan who actually introduced us to the system. And you can see this thing is snapping its claw. Why is it snapping its claw? We actually don't know. These males actually typically have guard a number of females that don't have the big claws, so it could be about signaling and territoriality. But let's do one more intuition here on these critters. So that's a real-time video taken with somebody's nice little handheld camera. And here I've slowed it down. And I'm going to tell you in a second the speed that our high-speed video cameras use to capture this motion. But before that, I'd like to probe what you know about imaging. So if you've taken a video with your home video or your phone or your iPhone, how fast is the frame rate that you can film at? So super cool numbers coming in here. I can't wait to share this with all of you. And just while you're filling that in, I was giving a sort of specialist lecture at Brown University recently, and asked this question of the group that studies, high-speed bat flight and so forth. And I asked them, what was the highest speed video that they had ever taken? And the answer was 1,000 frames per second. Okay, that was sufficient to see all the intricacies of bat flight. So I'll share, I'm going to end the poll and then share it with you all so you could see, you know, actually good job. Most of you got it about right. Most people film at 60 frames per second. And some of us have fancy iPhones that go to 150, and I'm super impressed that some of you have 1,000 frames per second, but that's pretty high tech. What are we filming this little video of critter moving a little dactyl with a diameter of a hair? We are filming at 300,000 frames per second. This is totally invisible to us. And in fact, this work and this field and the things that I'm going to talk to you about today are discoveries that basically happened since the capabilities for imaging became available to biologists, but specifically in the sense of having the light sensitivity so that we could film these organisms without cooking them. Before that, we had streak cameras and far less sensitive film that we just couldn't even grab this without it being too hot under the lights. So it's actually discoveries around light sensitivity. So how do these animals fit in? Okay, hopefully that gave you a little sense of how tiny these animals are and how fast they are and the world that I'm about to show you. And just to tell you a little bit more about this one system before I move into the general principles of these organisms, these amphipods on the left-hand plot show where they sit in terms of mass and acceleration. And because this is a public lecture and I wanted to, we actually have a much more complete plot that's just not published yet. So ask me about it on email and I'll send it to you. But basically what these animals in this discovery and a bunch of others that are not on this plot show us is actually the left-hand upper, sort of left upper corner of movements in biology, unmatched as yet by engineering, that are the fastest, smallest movements that can be done over and over again. And along that left-hand boundary, there's blank white space, and that white space is filled by other faster organisms like the jellyfish stinger that self-destruct when they generate motions orders of magnitude higher acceleration than, for example, a bullet. So these systems, what we're studying right now is the boundary of being able to generate movements like that over and over again without self-destruction. So we're going to address how organisms do that. And on the right-hand plot is where we're headed. We're going to look at the extraordinary performance of trapjaw ants and mantis shrimp and some of the principles that they have revealed, and then move toward the world of engineering and some of the discoveries that are coming out now. So what was my point of this slightly long, but hopefully intuition building intro? It was to set the stage for these systems. The thing about the fastest movements on the planet, and that's tricky when I say things like the control of power, is that everybody wants to talk about these systems as if they are very high energy and very high power in the colloquial sense of like a bright light. But actually, the energy is very small. And what's very big is the rate at which energy is released. And what's even more amazing is how these organisms control energy flow. In the middle panel there, I got small, springy, and ultrafast. So these systems are all small. They use springs to propel their motion. They don't use muscles and they don't use motors. So this is all about materials. And by doing the control of energy flow is how they get to ultrafast. So I'll show you that. So to start with, I'm going to do one more fun intuition building exercise with showing you a new discovery that we had about organisms, getting some of you onto planet science and some of you happily gazing up at planet science if that's not your comfort zone. Then we'll dig into some more heavy hitter stuff about limits in these systems and then wrap up with some points about interdisciplinary work. Okay, so let's talk about energy and let's talk about how these systems can do it. This is a picture of a Gallmedge larva. And if you look up on the right-hand side of that ruler, you'll see a little orange blob. And the orange blob is about the size of a grain of rice. that little orange blob just jumped from where it is, where it landed, which is where it is right now, from the tip of the pen. So that little orange blob just jumped that distance. That little orange blob does not have any legs, and yet it jumped that distance. So orange blob is tiny, which means a small amount of energy. And if you do a nice sort of overlay frame by frame, and those of you who've been watching the Olympics have been seeing a lot of this, this is what this little wormy thing looks like. This is a larva that will eventually turn into a fly that flies away, okay? So you can see that it took about 110 milliseconds to jump that far. That was a pretty amazing jump, by the way, a little bit above average for them. Now we can slow this down and take a high-speed video of it. And you can see this animal jumping into the air, and you can see it has no legs. So how did it develop a rate of energy release sufficient to get it off the ground, especially since I started by telling you there's not a whole lot of energy going on here, because anything that's small doesn't have a lot of energy. What these animals have evolved is a very funky system, which is that they have a little protrusion that attaches between the head and the tail, and it sticks together. I'll show you it in a second, with little gecko hair type adhesive structures. And then they scooch their body together and eventually press and release that latch, and then launch themselves with stored elastic energy. What you're looking at right here is a small amount of energy being adjusted in terms of rate by contracting muscles, but then stretching the elastic body using a latch that controls the energy flow and the energy release. So I can break it down for you into even more sections here. The little worm that will soon fly away as a fly, no legs, rolls into a loop. Somehow, we don't know how, it creates some kind of transient hinge halfway along that makes this thing basically have a little leg launcher. And at some point, enough energy is stored to actually separate the adhesive parts between the head and the tail. And that's what allows them to release. This is the magic of small energy, high rate, and control of flow. These little hairs up on that top panel are rather like adhesive gecko hairs. Considering I'm talking to people from the Bay Area, I am sure you have heard of gecko hairs. They're teeny tiny hairs that are so tiny that they generate tactile adhesion, van der Waals adhesion, most likely. We don't know that for sure. This is a new discovery. But what's really neat is if you look at that ventral view, the bottom of this larva has patches of hair all along its body. And it has just simply evolved to use a couple patches just for the purposes of this jump, and to basically break through that in order to control the rate of energy flow. So this is an example to build your intuition, but also so that those of you who are not physicsy can get what I'm saying here about mechanical energy, mechanical power, and control of energy flow. These animals are small, they're springy, they're powering their motion with materials that are preloaded with energy. And they're fast because they are controlling the flow of energy through things like latches. So we have springs and latches. This little plant up here is showing the little molts of these larvae. These larvae actually inhabit galls, and then they go to the ground and they jump across the ground and then dig a hole and then pupate and then fly away. All right. So hopefully some of you are on Planet Science, but if you're not on Planet Science with me yet, at least you're having fun. Okay, so let's talk about limits and let's get serious here for a minute. All that was actually serious, but let's dig into the nitty-gritty. Some of you are listening to me talk about this, and some of you are going to intuit that I am just talking about archery. I'm going to put that to rest in a minute, but yeah, we are in many ways talking about archery. So in archery, you have a muscle and you have an arm, and you could pick up that arrow and you could try to throw it and it would not get very far, and it sure as heck would not take down a deer. But you can take that exact same arm muscle, and instead of throwing with it, you develop a lot of force and you load elastic energy into the springiness of the bow. And then you let the bow throw the arrow. what is going on here? And by the way, your fingers serve as latches. What is going on here? Why is this such a great system? The reason why it's a great system is it allows your muscle, the motor, to operate in a space where it can develop a lot of force slowly. And that slow development of force over a decent distance gives you work, and that work gets put in the spring. But the spring doesn't have all of the limitations in terms of trade-offs between force and velocity that your muscle has. It has a different set of trade-offs. And typically a spring can release energy over a shorter time period than your arm can develop that. But it's not just that. A crucial part of why archery works is that your arm does not have to accelerate itself in order to accelerate the arrow. In other words, the weight of your arm is now out of the picture, and you're just accelerating the arrow, which is very lightweight, which means that all the energy in your heavy arm is put into the spring, and then the spring is throwing the lightweight arrow. This is all about a little bit of energy being released quickly, and the control of energy flow through the dynamics of the muscle. The muscle has to match the spring, and your fingers have to manage the latch. This whole field, when I came in, this is absolutely true, was all about calculating the rate at which energy was released from these fast organisms. And as long as that rate was higher than what muscle could do, you could get a big fancy paper and your project was done. And typically no one would actually look, or very few people would actually look at how the spring worked, and even fewer people would find a latch. What I'm going to tell you about today is what happens if you dig into the springs and the latches. And in the process of doing that, especially with this interdisciplinary team that we now work with, we realized that this is actually a class of systems in biology and engineering that we now call latch-mediated spring actuation. So systems that are launched with springs, and the energy flow is mediated with latches. So once we realized this, we were able to formulate an interdisciplinary framework for systems that use springs and latches. But this framework revolved around doing a few funny little things in physics. It required basically looking at the fact that these systems are small in biology. They're all small in order to get incredibly high rate energy release. And they're all driven by preloaded springs. The springs themselves are pushing and generating the motion, not the underlying motor or the underlying muscle. So we started to do some basic physics models, and we made this realization that actually at the size of these small organisms, springs have mass and their inertia actually matters. So for those of you who like physics, it means that these systems are not Hookean springs. It means that all of a sudden the spring itself matters. Oh my goodness, why did that just happen? No big deal, we're back. Okay, so what's going on here is that we developed this simple model. We scaled it down to the relevant physics of systems at this size, and we found a mass transition, which is that red circle surrounding the blue dot, below which it is better to use a spring than a muscle or a motor. And you'll see that there's a dashed line that says the ideal spring and the blue line that shows the real spring. But what's really cool there is that, again, up until this point, we'd always used sort of ideal systems, the perfect latch that releases instantaneously, or a spring that has no mass, that follows Hookean rules, that's linear. But if you step away from that, recognizing that these systems are tiny, it means that all of a sudden, these systems are very tunable. You might notice that I did not give you a big lineup of all the stats on all the fastest organisms on the planet, who's fastest and who's slowest, because today, what I want to do is instead point out that these systems can do a lot of different things. And it's not just about how fast, what the velocity is or the acceleration. So this plot that I showed on the right for my fellow physics geeks who are in the audience today is showing that transition, what we believe is now sort of the universal physics mass transition that we're now studying in biology, To the left of it is the space and the mass where you might prefer to use a spring or might evolve a spring. But depending on what you're using it for example, that top panel shows projectile takeoff velocity or takeoff duration or max power delivered to the projectile or projectile max kinetic energy. The mass transition is in a different location and the relative benefit of using a spring or a motor differs. Why am I harping on this? I'm harping on this for a couple of reasons. Incorporating size matters a lot. Throwing away assumptions that only relate to large systems matters a lot. It also matters a lot because biological systems are very diverse in this space and they're being used for all kinds of stuff, like stinging you while you're swimming. And that makes a lot more sense than an idealized system where we're just always talking about the maximum. So once we made this realization, it really opened up the field. And I won't talk about this today, but one of the big things that we're working on now is how a physics-based transition in mechanism it emerges across biology. So I'll talk about our mantis shrimp. This is obviously what I'm best known for, is our initial discoveries and sort of putting the whole world of fast movements on the planet out to the universe, starting with the mantis shrimp. We'll come back to this in a little bit. But this is actually a study showing how through evolution and through development, organisms can shift to a size where it is better to use muscle than spring. And so we can study this physics-based transition in whether to use a spring for propulsion or a motor or a muscle, and look at how biology explores it. But of course, whenever you're talking about materials and whenever you're talking about mechanisms and whenever you're talking about things when biology can do it and engineering can't, it starts to bring in a lot of people who are very interested in building these systems. So one of the things that happens when people start to come in and wanting to build systems is they start to ask you as a biologist, me as a biologist, how do they work? How does it work? So I'm going to tell you a little story about trapjaw ants. So trapjaw ants are up in the very top left-hand corner of that graph that's sitting up there. Trapjaw ants are one of the fastest moving organisms on the planet. And the way they develop this is by rapidly slamming their jaws closed. Engineers were really curious to learn about how this works. Now, I thought I knew how it worked until the engineers tried to start building one and it didn't work based on what we thought how it worked. So this was a great place to intersect. And it's also a wonderful example of how scaling matters in these systems. So I'm going to tell you a very short story about how trapjaw ants deal with a little bit of energy, rate and flow, and how they're small, springing, and ultra fast, and how this has come right back around to thinking differently about our engineered systems. So I showed you this picture of the archers, and some of you probably are like, well, dude, archery has been around for thousands of years. Why in the world are you studying these systems? We already know everything. But that big purple arrow is showing an ant on the archer's leg. We're back to the small business, okay? And let me flip it around the other way. This ant has to do what the archer does with its head. And somehow or other has to get all that rate and energy flow taken care of in its head in order to close those jaws. So that's a pretty serious space constraint for the bow and arrow system. So they don't do it that way. They do have a spring and they do have a latch, but it doesn't work like archery. So trapjaw ants, as I said, use a spring latch system, but they can also just close the jaws with muscle. Almost all of these organisms, the animals in particular, retain the ability to do a slow movement directly with the muscle and then switch over into the spring latch system. So these animals, like many of them, have both options. So this means that this isn't even just about size. It can just be about switching mode of operation. But really, we were curious mostly about the spring-driven strikes. How are these animals closing their jaws? We studied this really intensively. These are films. I'll show you in a second what it looks like. But one of the things that really boggled our mind is when we tracked these jaws closing in a really big data set. we found that they were highly consistent, so consistent that if you take the derivative of the motion, the acceleration is incredibly clean. And those of you who study motion, that rarely happens. That means that these are very precise, very consistent movements. And then once they pass the midline, they're all wobbly. And not only that, they seem to be rotating around a fixed pivot point. Why am I getting on about this? The reason I'm getting on about this is because of this question of where is the archer in the ant? So it turns out ants use their heads as springs. So if you look carefully, I'm going to switch between a couple stills here. But the first on the left-hand side, the picture of the ant kind of shows the ant's heads are puckering in from the side. And when I switch to when the jaws are closed, you can see the head bow back out again. All right, so it's puckering and then recoiling. And then you can see that happening again on the other one. And then a picture of the jaws closing at 300,000 frames per second. So they store energy in their head by flexing the whole head. But the question remains, how do they deliver energy to their jaws in such an extraordinarily precise way? So they have latches that hold the jaws open as the muscles load energy into the head capsule. And what we discovered is that these ants use what we call now a dual spring force couple. On the left-hand side there, it shows unloaded, which basically shows a lever with a muscle that can contract and open the jaw. And right next to it is the head capsule. They can lock the system closed. And then when they contract the muscle and the jaw isn't able to close, it causes the tendon on the muscle, called an apodeme, to stretch and the head to compress. Once they do that and release the latch, it means that on that right-hand panel, the recoiling spring pulls and the head pushes. Why is this interesting? This is interesting for two reasons, probably more than that, but I'll tell you two. One reason it's interesting is that these animals are able to close their jaws with the highest rotation rates of any system recorded, like orders of magnitude higher than any MEMS system that's yet been developed in engineering, without falling apart. And the centrifugal forces on this spin are so substantial that the jaw should fall off or the friction should stop the jaw from moving. This is essentially a mechanism for very unconstrained joint with a highly precise pivot point, which is what we saw in the earlier data. But even cooler, it is how the ant fits the archer in the head. In order to store enough energy with a strip of tendon, or the typical sort of tendony big long thing that we think about with springs, there is not enough space in the ant head. There's literally, if you tried to stretch something to store this much energy, it is bigger than the ant's head. So what you're seeing is the compression of a three-dimensional structure where all the deformations, tiny deformations, are spread over the whole three-dimensional structure to store enough elastic energy to fire those jaws closed. This is very interesting. This is how the archer fits in the ant head, basically. But it suddenly explains a whole lot of stuff that we did not understand before this about a lot of these systems. And it turns out most of these systems are deforming three-dimensional structures and using dual spring force couples to reduce the constraints on a system that's rotating at like 100,000 hertz. This is also super cool for building robots. And I'll come back to this in a little bit, and other systems, highly controlled, highly precise, highly rotating systems, where you're essentially using very precise deformation of three-dimensional structures in order to develop these types of motion, energy control. Okay, let's talk about the mantis shrimp for a few minutes, because I know that many of you are my mantis shrimp buddies. This is where, of course, where all of our work started, was putting mantis shrimp on the map. And from this flowed a lot of work and basically opened up this whole field. The right-hand side here is a picture of a mantis shrimp that is breaking open a snail shell with little hammers. Each of those hammers weigh the mass of 2 toothpicks, and they're moving with the acceleration of the bullet in a gun. I cannot break that snail shell without using a hammer. In fact, I had a pretty hilarious interlude in my lab recently where people have heard me say this a million times, but they had never actually tried to break one. And there they were with a big hammer, unable to break the snail. It's really hard to break a snail. And yet this little critter's doing it. How do they do it? They do it with the spring and the latch. They do it by deforming a whole external structure, little bits here and there, and then generating torque. But the reason why I'm bringing up this example right now is about this point that I started with, which is the control of energy flow. And I'm going to come back to this in a minute. But essentially, if you look up at that top row, you can see at the very far right, it says percent duration. That's the duration that it takes to load energy into the spring. Then if you go down a row, you can see that about 8% of the time is when the latch is releasing. 0.2% of the time is when the spring is actually pushing releasing all the energy and pushing the appendage to close and hammer. Then it rotates to the target in a millisecond, and then the hammer hits the target in 49 microseconds. Then the cavitation forms. Cavitation is that bright bubble you saw a moment ago, where the motion is so fast that the water actually vaporizes and it collapses in what's called the cavitation bubble collapse, which is arguably one of the most energetic natural events on the planet. And the reason why it is one of the most studied energetic events on the planet is because the energy is released over such a tiny period of time. This is a cascade of energy flow that these animals are able to use over the life of the system and for tens of thousands of strikes. This is a highly repeatable compression of energy release. What does this let them do? It lets them generate extraordinarily high impact forces. This is the left-hand panel showing impact cavitation of the hammers as they're hammering on a structure. And if you look to the right, that's a horn shark that also eats snails. And you look at the area under these curves and you look at the fact that the x-axis is microsecond versus millisecond. What you can see is that the mantis shrimp is a very, very, very tiny amount of energy system that is leveraging rate in order to do things that big animals can do. But we can reframe that, and this is our last little post for the group today to answer. We can line up where mantis shrimp are on this plot and compare the peak force that a mantis shrimp can produce to the bite forces of other animals. So if we could put the poll up, that would be great. And my question to you is, if you go across to the right along the x-axis in terms of mass, which of the animal's bite forces is the mantis shrimp's peak force for that hammer blow? That over to the right there. Yeah, the answers are coming in and you all are doing great. Okay, I'm going to end it. You all did really well, and I'm glad there's some good diversity in the answers here. I'm going to end the poll and then share the results with you all. Yeah, the peak force of a mantis shrimp strike is equivalent to the peak bite force of a hyena. Why am I bringing this up? I'm bringing this up because what we're seeing here is extraordinary control of energy release in a system that uses very little energy to begin with, and an alternative strategy for breaking hard things. So it's a much more complex and interesting question, I think, than what's the fastest creature on the planet. I feel like it's an interesting question that relates to a lot of issues related to size, materials, ecology. But let's look at this one more time with our awesome mantis shrimp. So here is the extreme cascade of energy flow and control. This is the difference in terms of orders of magnitude between 1 year and 1 second, or the difference in size between our planet and one of us. These organisms, these tiny, fastest creatures, have evolved extraordinary control of energy release. This cascade is all about compressing the time it takes to release energy. And the mantis shrimp remain close to my heart because they got me started on all this stuff as a pretty extraordinary mechanism to generate cavitation and peak impact forces. Okay. So let's briefly talk about interdisciplinary stuff, and then I can't wait to get some questions for you all. So once we have a space like this, where at the start, it's just, 20 years ago, it was, could we even film a mantis shrimp, or what is its velocity? That was the achievement. Or the achievement was, it produces more energy at a rate that mussels can't do. So therefore, they can't be doing this with mussels. And that was where we stopped. Now, 20 years later, especially in the past 5 years with the team that I've been working with, we now barely even start at the question of what's the velocity and acceleration, or how much power can they put out. In fact, really where we spend all of our time now is around materials and mechanisms. How do you control the release of energy without breaking things at these scales? What do these springs look like? How do you develop torque at 100,000 hertz without breaking the joint? This is a problem for MEMS design. And then, it goes on and on and on from there. So now these days, we are still working hard on discovery of these systems and populating those plots. And as I said, we have a much more populated one if you want to email me, to show some of the scaling around the lower limits of what is possible in terms of repeated use movement. But we also have a lot of robots and physical systems that are at least approaching what we can do, what we see in biology. But honestly, the part that I am most excited about is the materials and mechanical interactions. And so we work closely with materials scientists who can build actually these types of three-dimensional structures and these novel materials. I want to comment, though, that even with everything I've just said about how these mechanisms work and how fast we can film these systems, 300,000 frames per second, I will tell you that we still have never seen or resolved the release of energy from these structures. We can resolve the loading of energy, and we can do all the math in the universe to calculate energy in and energy out. But still, at these scales of an archer and an ant head, you cannot see with today's visualization abilities, or at least not yet in my lab, although I just got a camera that might let us do it. We have not yet visualized the material control of energy flow and release for precise motion. I want to end with just a fun comment here about a paper we just published. This is with Rob Wood's lab at Harvard about the mantis shrimp. And we built a robotic mantis shrimp. And, some people will love robots. I personally love physical models because physical models let us test questions about biology, as we had to do with the trap giant ant, is completely revisit how they control and release energy. Well, in the process of going and working with these incredibly talented engineers on this project, we made another discovery that I want to end with. So the mantis shrimp do everything that we've talked about so far, but the thing that I left out is geometry. And it turns out that we have been talking about latches and mantis shrimp as little sort of hooks and things that sort of brace against an interior and then release. But when we've studied these things, we've found that they sort of brace the system to allow it to be loaded and then let the spring push it out. But there's always been this delay between when the physical latch is removed and when the motion starts. And in this paper and in this study, we were finally able to solve a mystery about how is energy actually released in the mantis shrimp so that it doesn't tear up this physical latch. And it turns out that these animals, and of course the robot that they built, uses the geometry of the system to again control energy release. So even the tiniest tweak of a linkage system allows a system to stay locked and then briefly over center and then rotate out. And through evolution, the mantis shrimp have actually varied the dimensions of the geometry to adjust how much energy is released over a given time. So here we come all the way back to the foundations of physics. And one of the most tunable things you could come up with in terms of biological evolution, which is just relative lengths of links. So I hope today this was fun for some of you and a deep dive for others, and most importantly, made you curious to understand how fundamentally different it is to consider a cheetah running, certainly, and will always be iconic, fast movement. And how when we move into the actual fastest creatures, we move into this very diverse world of function, but also a very counterintuitive one that is so tiny amounts of energy, but with an incredible ability to teach us both on the biology side, the engineering side, the physics side, and neuroscience, by the way, because a lot of this is faster than neurons can process. The ability to control energy flow without destruction in systems that are lightweight and recyclable, and biodegradable. This is really at the heart of what biology is all about, and also at the heart of many of the things that we're trying to achieve in engineering. So in my introduction, it was meant that I have one other great passion, and I can't ever give a talk without mentioning it. I talked about being on Planet Science, and I know a lot of people want to be on Planet Science. And so for more than a decade now, I've worked really hard on the development of a software app and also programs that are running at multiple universities now to open up academic research so that it's equitable, accessible, and transparent, also to encourage interdisciplinary engagement. So ask me about that, and I'm happy to go on a big, long sidetrack about how important this is for doing great science and involving everybody who wants to be involved. So I would just like to end for just a second and thank the amazing people I've worked with, especially the members of my lab, but also this interdisciplinary team that I talked about with the 5 universities and my kids and all kinds of folks. Those left-hand pictures were a very happy picture where our team finally got together again in person with and without masks outside. And we've been working together for almost 6 years now. And you can see their names here, largely folks at UC Irvine, Carnegie Mellon, UMass Amherst, Harvard, and University of Lincoln. So with that, I hope that was fun, and I'm excited to hear some questions, and hopefully this was exciting for all of you to see what's new in addition to the mantis shrimp lab.
Awesome. Thank you so much, Sheila. Like she said, we can start taking questions now. I already see a few already showing up in the Q&A. So if you guys do have questions, feel free to go down to the bottom of the screen, and there's the Q&A button, and you can ask your questions right there. So we'll just get started right from the top. Fun one actually here. This first one from Scott says, aren't these movements supersonic? Are these like mini sonic booms?
That's a really great question. Whenever you're dealing with things that are this fast, we do get into the world of sonic booms. We get into the world of the speed of sound through materials or wave speeds. These tiny critters are typically reaching extremely high accelerations, but not the velocities that we typically associate with the sonic boom. But I will say, as an aside, this whole business of using cavitation by the mantis shrimp, and actually we also work on snapping shrimp. I mean, there's a whole menagerie of stuff I could talk about, but a bunch of these animals use cavitation. And cavitation, as I mentioned, is hugely energetic. And when the cavitation bubbles collapse, it emits heat and light equivalent to the surface of the sun and sound. And anything going that fast in water will eventually cavitate. And it is a sound barrier. So if you go above the cavitation barrier, you're going to make a lot of sound. And so a lot of like ship design and propeller design and stealth type of design, and even fish speed evolution is below the cavitation limit in order to be quiet. And in fact, the fish catching mantis shrimp do not cavitate, and I suspect it's because they need to be quiet. So actually the cavitation thing is a biggie around avoiding the noise that goes with extraordinarily fast motion. It's a cool question.
Yeah, that was a great question to kick it off. Very interesting. We have another one here. It says, at some point, you limit your description of creatures. What about the time it takes for a virus to latch onto a cell?
Oh gosh. Okay. So the world. Okay. So that's a really good question, and I haven't thought about this for a while. But we some of you may know Manu Prakash's lab at Stanford, and we worked together for a little while. But he's and his colleagues and Gilpin and others have worked on single-celled organisms and smaller, and how they move extremely fast. When you go down to that size and smaller, of course, virus and molecules and so forth, you move into a very different world, which is the world of high Reynolds number and highly viscous fluids. And the strategies get really different. So there's a really cool paper out of the Prakash lab showing how basically these single-celled organisms are jumping through the water, and essentially they're speeding up in order to get the water to feel less honey-like and basically less viscous. If we go even further down into the molecule level, there's a whole world of latches and energy storage. And at some point in my career, I would like to have a grand theory of fast movement and latching that goes from submolecule up to black holes. But for the moment, we're just doing small creatures because there's so much to learn right there. But that's, yes, don't even get me started.
That's amazing. Yeah, that I'm sure it can, you'd want to scale all the way up and all the way down if possible.
Yeah.
Unified. Yeah, that's awesome.
It's all about energy control.
Yeah, energy is everything. Another one here, it says, it's a little bit of a long one, but I'll try to lay it out here. It says, what are the implications of the Gallmedge larvae launching itself for potential physical therapy practices for individuals who need to get body parts like legs, arms, or other moving and working again after injuries? Is there any science to be shared or used to help in the physical therapy context?
All right, that's a great question. In fact, one of my collaborators, Greg Sawicki, who's now at Georgia Tech, works on exactly this question, which relates to how can you use elastic structures in order to, for example, reduce the amount of energy someone needs to use to walk, or to make a system more efficient, or to make a prosthetic more lightweight. So absolutely, our work connects into a lot of these different scales about how do you strategically use materials to shuttle energy in a way where you might want to have energy in a more efficient pathway, or to reduce damage, or in some cases to actually store energy and release it to enhance motion. The one thing I will say about that world is that it's at this larger scale. It's larger than the systems that we're studying. And in fact, it's very well studied. It's beautifully studied. So when we intersect with those folks, which we do all the time, that's the world of elastic systems that are very heavily involved in physical therapy and all sorts of therapeutic devices, we're often coming together around how do you use three-dimensional structures? How do you navigate energy flow? What are the materials that we're using? And then we're trying to tug that down into this even tinier world. Great question. But yeah, the world of elastic mechanisms is a big one for human health.
Yeah, that's super interesting if it could be applied to things such as getting better from an injury, right? Mm-hmm. Another question here, it says, how does a spring latch system like this evolve?
Oh, I love that. So, I mentioned at the beginning of the talk that this type of work invokes neuroscience, evolution, all of which we have studied in my lab. And in fact, I part of me was in the mood to give an entire talk about evolution this evening, but I went for a slightly different one. Have me back next year and I'll give you an evolution talk. So the evolution of these systems is super interesting. And, the origins, why organisms actually evolve these systems is a really interesting question. I think that there are a number of reasons why organisms evolved these spring latch systems. One of them relates to temperature independence. So for animals, especially things like insects, the temperature of the environment drastically affects the ability of muscles to contract. So a cold insect, for example, or a cold snake for that matter, the muscles will contract very slowly. But when they do that, and the chameleon tongue is a great example of this, they can still very slowly load energy into a spring, and then the spring can operate in a way that's independent of the ambient temperature. So one hypothesis is this relates to thermal independence, and we're actually looking at in the lab from the other direction, which is as the oceans warm, are these animals that use these spring-based mechanisms like snapping shrimp, different than mantis shrimp, actually protected from climate change in that way? Or actually, are they more vulnerable because the actual material properties for these springs are affected by things like ocean acidification? So that's one answer. The other answer comes back to this multifunctionality business that I was talking about with the trap jaw ants, where they can close the jaws with the muscles, but they can also switch over to loading their whole head with elastic energy storage. And what we see over and over again in the arthropods, like the insects and the trap jaw ants and the mantis shrimp, is the retention of the ability to move with muscle, and then taking the muscle that counteracts the extensor muscle, so the antagonist, and evolving some kind of latch structure there to brace the system to not be able to move until elastic energy is stored, and then the release. So it seems to just use these antagonistic muscle pairs and then making one the spring loader and the other the latch controller. And that's the pattern that we see many times. So that's another way to answer that question. I would love it if the world would start working on the evolutionary origins of spring latch systems.
Yeah, we'll definitely have to have you back on. I see a few comments in here saying, we'd love to have you back and talk a little bit more about the evolution side of things. Too many topics, too little time. So hopefully we can have you back again soon. Another person here was asking, very interested in the presentation. I wonder if you would be willing to share the slides. We could share it with the audience, or is this not a shareable presentation?
Oh, yeah. Oh, the slides from today?
Yeah,
Oh,
from today.
sure. Yeah. You are welcome to all of those. And then if anybody wanted to see the more completely filled graph showing the limits of repeatable motion, just email me, because it's actually my graduate student Jacob Harrison is finishing up his PhD, and it's sort of like this great graph. And I have a rule that I don't scoop my own students and let them show it to the universe first, but it's pretty cool. But they're all the slides that I showed today are for the universe. So anybody can grab them. I'm also happy to.
We'll plan to get those from you, and audience members will send it out in the follow-up email along with the replay link, so you guys can rewatch this and check it out. So for those of you asking in the chat or in the Q&A, you can do that. Okay, terrific. Next question here says, how large can an organism be before this type of system is no longer feasible?
Oh, I love that question. So we currently have a grant from the National Science Foundation to basically probe that question using the diversity of systems that we know are using spring latch systems. Now, I will say that because of the history of this field that tended to sort of black box what was happening with the spring system, it's still like constant discovery. Like I feel like every day I open up a journal and find somebody new has discovered some cool system that's using spring latch mechanisms. But essentially the mass transition that we put up on that physics plot is approximately where we predict that transition to be in biology. Now, the thing is, biology is always very clever and very diverse. And I had a great question recently that relates to this, which is this mass transition relates to the thing being propelled. In other words, the analog to the arrow. And biological systems are very clever with having a small body part doing the fast movement and not necessarily the whole body. So one of the cool ways to ask where that mass transition shows up is to look at, for example, insects, and Greg Sutton at University of Lincoln has done this, where insects are propelling their whole body. And in fact, trap jaw ants not only capture prey with their jaws, they actually jump with their jaws. That's evolved a few separate times too. So, but the insects are particularly cool because a lot of jumping insects store elastic energy, but the bigger they get, eventually they switch over to using muscle. So the mass transition is more easily studied in systems where they have to launch their whole body because they hit that upper mass limit pretty quickly. Frogs are also very cool with this. So those are some of the ways that we can tackle this. And of course, I have a dream that this will be sort of a new principle for biology in terms of locomotor or motion transitions, this mass transition. And I probably should say that it's not just animals. So a graduate student in my lab, Justin Jorge, studies plants. And plants also use spring latch systems, and they also have to navigate this size-based transition in terms of the way they're generating and storing this energy. So this is pretty cool. And actually fungi. I could give a talk on fungi. So those are also very cool. So yeah, we could see it across the tree of life, and I'm pretty excited to look at how that's happened through evolutionary history.
That's amazing. Yeah, we actually do have another question kind of related to that. It seems pretty similar, but I think it has a little different connotation. But it says, aren't the physics here fundamentally size limited? That is, understanding the biology is fascinating, but it still doesn't allow you to scale up the mechanical delivery system to a size that's useful, right?
Okay, so really good question. And actually it's a very important question. And you know, when we first were working on this system on the interdisciplinary team that I've mentioned, the multi-university team, you know, at first I thought this was a big problem where I was like, oh no. In fact, my mom was visiting this weekend. She's a retired language teacher, high school teacher. And she's like, well, can't you just do a 3D print of a great big ant head and solve your imaging problem? And I was like, well, that is a really good suggestion. The problem is the physics just doesn't, like, the physics are the same, except that when you scale down, you can actually get the appropriate amount of energy storage, and you can be moving something small enough that using springs works. So at first I thought that was a problem, that we couldn't scale it up. But on the other hand, what I'm realizing is that a lot of the engineering invention and world and focus right now is actually on really small things. Things like, you know, one of the projects that we did with the fungi fire their spores with the surface tension energy of a droplet. That ended up being an awesome collaboration with an engineer that uses jumping droplets to both clean electronics and take heat off of electronics. The other place where we see these tiny little things are all of the MEMS system, micro electric mechanical systems, MEMS, that are doing all kinds of jobs. So actually a lot of electronics and engineered systems are moving smaller and smaller and smaller and hit the limits that these organisms have gotten around. So you might not be interested in robotics, I get it, but there are other parts and pieces to this that are really crucial, and a lot of engineers have already recognized this. So it's essentially about energy control and precise, very rapid motion using these sort of basic components and leveraging the inertia of these tiny systems in order to do this type of motion. Another really cool one is puncture, and actually being able to do targeted drug delivery where you can actually puncture into individual cells and so forth with highly precise pre-allocated tiny amounts of energy. So yeah, at first I had the same kind of knee-jerk thing where I was like, oh, bummer, I can't make a great big ant head that moves well, maybe I'm glad, maybe I'm relieved that we can't have great big ant heads moving, you know, the 10 times the acceleration of a bullet. But no, it's going to have to be small. Anyway, yeah, great question. Really, really good point.
Yeah, we're getting a lot of great questions in the chat, and they just keep coming. So another one here says, is the crossover point of trigger mediated and muscle powered mechanisms in animals likely to apply to engineered systems too? Or do you think engineers will be able to shift to that point in direction of larger systems?
Okay, yeah. So this crossover point for mass and for latching is definitely relevant for engineers. And after having worked so closely with engineers in this space, what I'm finding is that any novel pathways for controlling energy flow and reducing time and doing it in a way that doesn't break the system is pretty much the name of the game. So we are constantly coming back. And, you know, when I work with the engineers who are fabricating these small systems, what I hear all the time is the limits of the materials. The materials are breaking, they're falling apart, they're tearing the system, you know. So actually there's a lot there around these materials not failing and how to basically take these systems all the way to the limit without breaking. On that note, I did want to say I was talking about this stuff the other day with another group, and they actually brought up a really cool point about the latch system and the Gallmedge larvae. And I kept going on and on about how, you know, it's repeatable, they can jump many, many times. But one of the people at the seminar pointed out that actually when you're doing a latch like the Gallmedge larvae with the adhesive little hairs gripping onto each other, and then they suddenly break apart, that's actually an example of failure, but that can be reattached. So there's also a really fun space here for thinking about taking materials all the way to the limit in order to achieve this power, sort of amplification of mechanical power, but then still being able to reload and rebuild the system. Anyway, lots of classic questions that biology systems have to navigate, and then engineers come into from a different lens, but same problem.
That's amazing. Another question here from an AP biology student in high school. Can you touch again on how evolution has helped develop these amazing mechanisms?
Yeah, totally. And actually, I'll tell you a different story this time. This would be another one I'd tell you if I was doing a whole talk about evolution. So we've done a lot of work. Actually, one of my passions, I mean, I'm talking a lot about applications because I was thinking like, you know, Palo Alto, Bay Area, it's going to be about engineering. But, you know, the other part of this whole thing is the massive trade-offs that come along with these systems. So we have worked, I mean, one of the big areas for my lab is studying the intersection of evolution and physics. So evolutionary processes are totally different than how engineers typically think of, you know, biology as being the sort of these perfect devices, and they really aren't. They're a product of all these trade-offs of like what you get, what you lose when you're moving through this space, and they work reasonably well. So one of the things that we discovered when studying the evolution of the mantis shrimp, so there are about 500 species of mantis shrimp, and we have spearers and smashers and all kinds of other stuff. What we found is about 50 million years ago, when the smashers evolved, and they switched over to being incredible, like much faster. They have a more intense spring latch system and all kinds of other stuff. We found that the components, when they vary and when they evolved across species, the components always basically changed together. In other words, they started to be more correlated and tightly integrated. And when that happened, the rate of evolution of sort of the accumulation of different morphologies slowed down. So what we found is that the switch to these super tightly integrated and tuned, sort of cutting edge, extreme systems reduced the ability of these groups to evolve more diverse mechanisms and structures, whereas the slower mantis shrimp with slightly less integrated systems were able to diversify much more substantially. So there's really cool stuff around this intersection of physics and evolution operating at extremes that I just love. So really, really great question. Thank you for that.
Yeah, of course. And this obviously shows we do need to do another one at some point because you could talk for hours on it, I'm sure.
Oh yeah.
I have a question here. I hope you know what he's referring to, but it says, how does your work help to explain the hot spit described by Tom Eisner at Cornell University?
Yeah, great question. So Tom Eisner is one of the greats in this world of organismal biology. And I believe the question is relating to some of his discoveries that are essentially insects that take two chemicals and mix them together and cause explosions, like fiery, like burning explosions. So yeah, so I guess nothing would surprise, I guess, given the level of geekness that I embody, it would not surprise any of you that I have agonized about this question of what does it mean to be able to do something many times as opposed to something that's an explosion or repeated use. So I decided to bin organisms that basically set off explosions, but don't, self-destruct the whole system in a different category than these systems where they're essentially staying internally, like the mantis shrimp, nothing's breaking and nothing's exploding, and they're able to still load all that energy in and control the output. So we put that in the combustion bin, which is a little bit different. But I think it's a really, really good point. And so when we do these kind of bullet type comparisons, really the bullets are doing the more of the combustion option of where you're using like an extreme release of energy to fire it. In the case of the bullet's gone. In the case of bombardier beetles, the beetle's still fine. It just set the explosion off behind it. So really cool question. And yeah, love that stuff. I haven't ever worked on it though. Maybe I should look into that.
Yes, this is amazing. So many different topics, so many great questions. This is a very interesting one. The creatures you described seem to be largely underwater dwellers. Is there a reason these high-speed motions tend to be more prevalent in aqueous environments?
Oh, that's an interesting question. I think that I'm just kind of biased. So, on my list of like things I wanted to do with my life, music, long distance sports, I also wanted to be a marine biologist. And so I had this like leaning, I always have a leaning towards looking in the ocean for things. But the truth is these systems are found in every habitat. And so I just happen to emphasize more marine systems. That's what I just gravitate towards that. But there must, I feel I probably somebody in this audience actually knows a real number of how many species of jumping insects there are on land that use spring latch systems. It's got to be thousands probably that do that. So actually they're very prevalent in both habitats. And I think the thing that's so cool about the underwater systems, there's also bladderwort plants that do this stuff, is this piece with the cavitation and the sort of speed limits and flow issues when you're in water. So I get also pulled into that because I find that really, really interesting because it adds a layer into sort of limits of do you cavitate or not, but also has led to some systems that have evolved to really tightly control the presence and absence of these extreme fluid dynamic events. So I am drawn towards that. But yeah, that's just a bias of me talking today, basically.
Yeah, that's interesting. I think I would have thought it was in aqueous environments, but like you said, it's in every ecosystem, which is amazing. Question, kind of a double question here. It says, how does the saddle-shaped structure of mantis shrimp withstand continuous deformation and reformation? And as a follow-up to that question above, does that saddle-shaped structure regenerate just like the club structures do in response to cavitation?
Oh, great question. Boy, there's someone who knows their mantis shrimp factoids. Yeah, I didn't even talk about this. So when I talk about these three-dimensional structures changing shape, one of the iconic three-dimensional structures is in the mantis shrimp, and it looks like a saddle, like literally a saddle on a horse. And what happens is actually the whole exoskeleton flexes when the mantis shrimp is loading that spring. And then in some species, the saddle is a crucial part of storing elastic energy in addition to that and reducing failure. So from what we can tell, that elastic mechanism is able to be loaded like thousands of times. So we've put it on our materials testing machine and just tested, and it's just fine. Like for those of you who do material stuff, the extremely high resilience. Essentially, you can just load it for eons. So as long as you're loading it within its sort of safe region zone. It can do it many, many times. And yes, these animals molt, the mantis shrimp do. And typically where we see the wear and tear is on the hammer. It gets worn down. And I really, frankly, have never seen wear and tear on the actual elastic mechanism. But yeah, they shed the entire thing, grow a new system underneath, and then molt it, and it's fresh and ready to go again. I've never tested the elastic mechanisms throughout the whole molt period, but I think that would actually be really interesting to see how that shifts over time, or the through mineralization and things like that.
Interesting. Yeah, and there's actually another question kind of along the same lines. It says, can a mantis shrimp repair an injured limb, or is it impossible to reestablish control after some types of accidents or injuries?
So that's such a good question. I kind of feel like there's a resurgence of, or let's say I think we're starting a new era of discovery now about the ability of crustaceans to repair their bodies. If you're interested in this stuff, Ali Miseres in Singapore has done some phenomenal work on molting and mantis shrimp that really throws our assumptions, like at least of my generation, is that once they're done molting and they have their fresh exoskeleton in place, there's not much repair going on. But Miseres's work is really pointing towards this being a much more dynamic, real-time process. And I think that just remains to be seen. But, historically in the world of crustacean biology, we would assume that very little repair is done, and the repairs are done only during that molting and shedding time period.
Mm. Yeah, that makes sense. Another one here, it says, do trapjaw ants possess a saddle-shaped structure similar to the one behind the mantis shrimp clubs, or is this simply integrated into the ant's head?
Oh, what a great question. So I've worked with Andy Suarez's group at University of Illinois, Urbana-Champaign quite a lot. And his team has just incredible diversity of trapjaw ants that they've studied. In working with them and looking at the diversity of ant heads, I would say that the saddle shows up a lot in these head flexing mechanisms. So the Odontomechus barri, which is the one that I showed today, which is found here, Florida, actually it's just made its way up to North Carolina, it's all through Central America and so forth. That one has this sort of puckered inside that you could look at as a saddle, but there's some other species that do look very saddle-like. Yeah, and I'm really, one of the things that I'm just really excited to work on, we just got a grant to get a really, really even more ultra high speed video camera, is to try again to see how that energy is being released when the head flexes and pushes those jaw closed, because I think that's when we'll see more clearly whether there are particular regions of the head that are flexing, and whether we're seeing that saddle-like shape show up in terms of the concentration of where the energy is being stored and delivered. But yeah, that the saddle mystery continues.
That's awesome. I know we're running tight on time. We have a few more questions, so I'm going to try and jump through these. I apologize if I can't get to all of you, but I want to be cognizant of Sheila's time and everyone's time. It's kind of towards the end of the day. So I'll finish up with a few more here. One here says, how do cone snail radulae fit into the speed graph? Are they on the faster end of the spectrum?
Oh, that is such a good question. One of the colleagues on this interdisciplinary team I've talked about, Manny Azizi at UC Irvine out there near you all, has worked on this as well as his colleagues, and they have a beautiful paper in Current Biology on the cone snails. For those of you who don't know, the cone snails have basically a poison arrow mechanism that they, if you get one in your foot, you're in a whole lot of trouble. Of course, they normally want to just fire it towards a fish and catch the fish and eat it. And they basically store up energy and then squeeze behind their poisoned radula and then shoot it out. And it's tethered but they can also replace them. So it's actually more like it's almost a fluid-driven mechanism. So in terms of their speed, they're not super duper fast. Like they're definitely not sitting on that left-hand boundary that I was focusing on today with the amphipods and some of these other super tiny systems, but they're very respectable. And I will say, I feel like one of the reasons why they're a little bit slower is that the radula is kind of big, and that is often used for actually tethering the fish itself. So it's a slightly different job than, for example, the teeny tiny stinging cells of jellyfish. But yeah, if you haven't looked at the cone snail mechanism from that recent current biology paper from the Azizi lab and some colleagues, you should definitely check it out. It's really mind-blowing. But yeah, same principle, springs and latches.
Wow, that's amazing. Another question here says, can you speculate on a relative degree of evolution of the structural parts of our bodies, like bones and joints, in order to resist the potentially self-destructive force of the spring latch mechanism like what you described?
Yeah, so I'm currently teaching an animal physiology class to a bunch of future doctors, and I'm always talking about, they're not all doctors, they're interested in a lot of different things, but I often talk about this assumption that we come in with, that human bodies are sort of the pinnacle of everything. You know, when I think about, you know, bones and the human body size, and I try to place that in the context of the systems that we work on, it's a really different ball game, right? So we're big. We don't have an exoskeleton, so we don't have those types of spring mechanisms, although lots of therapeutic approaches for humans actually do involve literally attaching an exoskeleton, enhancing elastic processes, and, you know, reducing damage and so forth. So engineers put exoskeletons on humans. But really the one big thing that we have in common are our muscles. And our muscles are limited in the same way that a mantis shrimp's muscles are limited in terms of being able to develop a lot of force quickly. And that's why we have, as engineers through history, developed things like archery to do what other systems have evolved in their own bodies. For us to withstand the impacts of these spring latch systems, honestly, like if you get hammered by a mantis shrimp, it's really not so bad. What's really bad is so their hammer's basically on their mouth equivalent of an elbow, whatever you want to call that. But at the tip of it is a spear, a very, very sharp spine. And that, I got to tell you, is a bummer in terms of getting stabbed. So actually that's more of like just making sure you wear a wetsuit and wear gloves.
Awesome. All right, coming up on the last two questions here. One says, could vibrations induced in materials such as crystals by radio frequency stimulation create enough acceleration to compete with latched mechanisms?
So that's really interesting. I don't know. I think that's a really interesting question in terms of how do you get equivalent energetic control at the rates of these systems, and how do you amplify that? And that's a possibility. I will say, right, that people who study cavitation and want to control it and direct it are often talking about those types of energetic inputs and control, and how do you get controlled cavitation. This is a total, like, how shall I say, tangent of sorts, but I will say that the ability of those mantis shrimp with the sort of toothpick-sized mass hammers breaking open the snail shell, I've thought for a long time, and many engineers have shared this with me as well, that it's very likely that actually those impacts are setting up a vibration in the material that make it easier to break on the third or the fourth hammering strike. So it's not what's being asked, but just sort of flipping it on its head, that sometimes the development of these kind of waves and vibrations can change the material properties of something rigid like a snail shell that could make it quite brittle and easy to break. So there's a lot of pathways in here, but absolutely this issue of energy in materials is a biggie, and it has fingers, you know, in all sorts of different types of engineered and physics systems.
Yeah, that's really interesting. All right, we're coming up on the last question. I know we're at the half-hour mark and we're trying to finish up here, but I think this is a good one. Does cavitation occur in air, or does that phenomenon only occur in water? If so, how do trap-jaw ants, for example, cope with this? I've heard that ant teeth are coated with a layer of zinc atoms. Could this play a role?
Yeah, really cool question. I get that question a lot about cavitation because some people think cavitation happens with lightning and thunder. But again, there's water vapor typically around that, and I've never really been able to understand that. So probably somebody on this audience could probably explain it to me. But the cavitation issue specifically that we talked about, like with the mantis shrimp forming a cavitation bubble and having it collapse against the shell and generating these different force peaks, is something that basically these air living ants just don't deal with at all. However, you are absolutely right. Trapjaw ants and many of these arthropods, including the mantis shrimp, have an outer layer of highly mineralized, highly brittle structures in order to basically develop that hardness that goes along with these sort of high instantaneous peak forces. So that's definitely the case in trapjaw ants. It's actually true for termite jaws. That's why they can take down houses. I know that's a big issue out there in California. But the cool thing, just to add on, to just wrap this up, probably one of the coolest outcomes of our work on the mantis shrimp hammers relates to exactly that question. And again, this was done out in California, UC Riverside. A group there solved why the mantis shrimp hammer doesn't break. And in fact, what the mantis shrimp hammer has is that sort of highly mineralized layer on the outside, but because of the shape and the sort of plywood layering of the materials inside, they're able to channel that impact energy to the interior and then vibrate it out so it doesn't break the hammer itself. So there's some really cool stuff around even the structures that are being used, like the jaws of the trapjaw ant and the mantis shrimp, that allow them to not break it even while wielding these extraordinarily brief and extreme energy release events. So yeah, a whole other very, very cool topic. And by the way, that has been translated into materials that are used, helmets, in airplanes and all kinds of stuff. So that's a really nice translation that's come out of the mantis shrimp work.
Wow, that's so interesting. From mantis shrimp to helmets, like, who would have thought, right?
Who would have thought?
Awesome. All right. Well, I think we'll wrap up here. Thank you everyone for tuning in this evening. And thank you again, Sheila, for taking the time to speak with us tonight. This was amazing. I think the topic was super interesting for all of our attendees, and they definitely found it fascinating based on all these questions we had. For all of our audience members, the replay link will be sent out in a follow-up email, and our next Café Sci will be coming in March. So you can be on the lookout for that communication related to that event. And with that, have a great rest of your Thursday, and I look forward to seeing you all next month.
Thank you.